I would like to talk today about something completely different. Thank you very much, Praneeth, for your presentation, because I think the contrast between how advanced is the modeling and the little steps that we do as experimentalists is the actual problem in the field. And I will talk about this. So my presentation is titled Urease Autocatalysis, Dreamer's Blessing, Engineer's Curse. I will quickly introduce the topic, talk a little bit in general about what we do in the field of artificial enzymatic reaction networks and enzymatic oscillators, and basically guide you through my adventure with the urease oscillator, show you a urease photo control module, hopefully make some very interesting statements in the outlook. So the question that inspires us is, how do living systems compute? So this is one of my favorite videos in which bacteria swim towards the sugar crystal. So what is wonderful here is that bacteria is actually very small, and the difference in the concentrations of sugar at the front end and at the back end of the bacteria is very small. So bacteria actually has to measure the change of the concentration of sugar in time and determine the direction of swimming accordingly. Bacteria is very small and doesn't have a lot of resources to do so. Plants, we all know that they can find the way towards light in very complicated environments. So as chemists, we are very inspired by this, and we would really like to create chemical systems that would have these opportunities of living, to sense, to integrate various stimuli, and to take decisions that are based on the signals from the environment. So how do we do that? We use this framework of thinking in which we separate inputs from the outputs. These can be molecular inputs and physical stimuli as well, such as the pH, the temperature, and the light. Wherever conditions we set goes into the reaction network. I hope you see my pointer, by the way.
We do.
Yeah, very good. Thank you. Because it will get more difficult with time. So in the reaction network, the concentrations of reagents, the concentrations of enzymes, in the case of enzymatic networks, set the reaction rates. The output of computation is the set of concentrations of the products. And in this case, we talk about biomolecular computing. However, if something happens in response to these processes, we can also have a functional output, such as movement, growth, whatever other function. And in this case, we use the same framework to talk about lifelike materials and to try to design them. Basically, enzymes are the molecules that are used by the living systems to create these functions. And me and my colleagues recently published this very fundamental review on artificial enzymatic networks and how to create all sorts of these emergent properties in there. I would like to focus on one particular aspect in this presentation, but I'll start in general. Basically, despite the networks that are found in living systems, they usually consist of hundreds of connected reactions, in some cases tens. And as I see from visiting the seminars, you actually have quite a lot of success in modeling of these networks with tens and hundreds of reactions. It appears that by now, the artificial networks that we can actually create in the lab, they mostly consist of a single nonlinear co-reaction, because it's basically close to the limit of what we can design rationally. And this is because we have the additional processes. So one reaction is not enough. We usually need the additional processes, one or many. This can be side reactions, transfer processes. This everything takes place in a certain type of reactor or setup that is very important. And basically, by more or less unknown design rules, it can give us this, what we want, complex nonlinear dynamic behavior. If we look at the sorts of co-reactions that are described in theory and that are employed in practice, we actually find that even here we have several mechanisms that are well known in theory, and some of them since 60s or 70s, however, never been realized in practice. So in practice of enzymatic networks, the two most common motifs is the proenzyme autocatalysis, in which an inactive precursor by the action of the product turns into product. So this is a classical example of autocatalysis. The examples are networks of the enzyme trypsin and chymotrypsin. I will show one example in the next slide. And I will talk more in this presentation about networks of urease, in which basically the product does not directly catalyze the reaction, but it enhances the rate on the enzyme. So the more P is produced, the more active enzyme becomes, then again, the more P is produced. So we also classified the additional processes that are used in such networks. There are not too many, but they're actually very practical. And I think with additional processes, we are better than with coupling of core processes. Thanks to classical biochemistry, because of the concept of enzyme inhibition, we can also make two enzymes work against each other. And there will be one example of that. The most interesting estimate here is transfer processes, because among chemical processes, these are the ones in which we have the least means of control. We don't have a direct connection between molecular structure that we do and the rates. Equally important are the types of reactor setup. So in non-steered, steered, batch, steered with inflow, and outflow, or in a thin layer, you can have very different dynamics. And again, I will have examples about that. So a little bit on enzymatic oscillators. I think the BZ reaction is very well known among this group. And basically, the long-term ambition was to create an oscillator that is based on enzymatic reactions. There is actually one very well-studied oscillator, is the glycolysis oscillator in cells. Instead, we are talking here about purely artificial systems that I can assemble from pure regions in the lab. And so the first example that was discovered, I believe, in the end of 60s by Japanese scientists Yokota and Yamazaki, is basically an oxidation of NADH by the oxygen of air in the presence of the enzyme horseradish peroxidase. Works beautifully, and there is a lot of research on that. This reaction possesses a very rich phase diagram with many various states, including double oscillations, chaos, etc. The problem there is that it takes place in a reactor in which you should both add the reagent continuously and ventilate the chamber with a mixture of oxygen and air in a certain proportion. Sorry, nitrogen and air. I wish I would know how they actually discovered it, because the setup is immensely complex. So instead, a later example from my colleagues and predecessors in 2015, the trypsin oscillator has been designed rationally. And basically, it's using the trypsinogen autocatalytic loop modified with two additional feedback loops, produces oscillations in a continuously steered tank reactor. So there is a continuous inflow of reagents, continuous outflow of products. There were some follow-up papers about that. It's basically now the golden example of how you can design such systems rationally. And another system that's been long in development is the potential for urease oscillator. So I hope I will explain this clearly, because this slide is key to the understanding of everything that follows. So the molecule of urea is neutral. Basically, the enzyme urease catalyzes its hydrolysis, so reaction with water, to produce ammonia. Ammonia is a base. So as we start in an acidic medium, the ammonia produced increases the pH, and thereby it influences the activity of urea. Basically, we have the autocatalytic loop. The rate of the enzymatic reaction basically is described in this case by the classical Michaelis-Menten equation that is modified with this term that is responsible for the concentration of protons, the pH, in other words. And it contains these two constants that are responsible for ionization and deionization of residues in the active site. So what we should understand here that as it is acidic, the rate is low. As it's very basic, the rate is also low. Somewhere in the middle is very high. And as well as we increase the concentration of urea, the rate increases. So the whole dream here is that if we would find a process or a side reaction that would drive our state back from the alkaline at 8 to the acidic at 4, 6, we would be able to basically go by these sigmoid and cycles over and over again, so to create an oscillator. I hope it is a little bit clear at this stage. And as all actual knowledge, it's I think not published anywhere in these direct words, but among the requirements we need not only this autocatalytic positive feedback loop and reversibility that I'm talking about now, but we also need the two feedback loops to be delayed. So they would not simply come to the equilibrium state somewhere in the middle, but they need to compete against each other, never reaching the equilibrium. And in the following slides, I summarize a series of papers that are mostly done by colleagues and collaborators of Professor Annette Taylor from Britain. She and her team, they developed a whole theoretic understanding of systems with urease. Again, the reaction equation is just repeated together with the rate equation here. What these models also take into account are the acid-base equilibria for ammonia, hydrocarbonate, carbonate, and water. Basically, they characterize how the production of ammonia influence the pH in the system, and thereby, by these terms, they influence back the activity of urease. And eventually, this the system in view is a system where urease is immobilized in a particle to which the protons and the urea can diffuse from the bulk of the solution. So it's a particle that is surrounded by the medium that has a certain acidity, a certain concentration of urea. So in these papers, they reduce the system to these two differential equations and analyze them in a great detail. I think everybody, but not me in this seminar, can understand how they do it, understand the general mechanism behind it, but not the future details. I very much recommend reading these papers. And basically, the conclusion here is that there must be a region, quite a narrow one, a region in the section of the phase diagram by the rate constants for the substrate and for the protons, where we should observe oscillations. I believe in that. So now, how to find these? The first issue that the chemist faces when tries to work with enzymes, although they're not that tricky, all of them, but particularly urease is very tricky because the activity of the free enzyme just drops in 1 to 2 hours. So it starts changing immediately as we prepare the water solution. What we did, normally with other enzymes, we prepared we mobilized them covalently on acrylamide microbeads that used to stabilize them very well. However, not for urease. The activity still dropped. We even had a plan B to use a urease from another source that potentially would be more stable. We found an alternative option to just physically entrap them without a covalent leakage. Apparently, the enzyme just didn't like covalent leakage. Anyways, we arrived at the state where we have stable particles, very high activity, and the activity doesn't change in weeks. So very good. Now, the system that we use for detection looks like this. We have these particles distributed in a cell. It's roughly 5 by 5 millimeter. So there is basically a drop of this reaction medium that contains acetic buffer and urea. So there is a transport urease reaction in the bit. We consider in all of our thinking that the rate of the reaction is slow enough so that the bulk of the medium does not change during the time that we observe. And this is how we see it in the microscope. So the beads are 50 micron. We put them sort of in the cell, and the fluorescent dye basically helps us to locate them, but also to understand what is the pH, whether it's acidic or basic in this place. And so the first thing that we have seen was actually very interesting, because it is already a sign that autocatalysis happens in this system. Because normally, if there would be sort of a usual chemical kinetics, you would see just a spread of the produced reagents. Instead, here we observe what literature calls reaction front wave. So we have a separate region in which the system is sort of reacted, and then it spreads over. This behavior is also a little bit similar to this very famous paper from Science, where the researchers put, I believe they put here the pieces of food on places where there are stations in the Tokyo subway. And basically this organism, the slime mold, it found the optimal way between these that was compared to, that was appeared to be very similar to the actual map of the Tokyo subway. So this behavior is very interesting by itself, but we are not satisfied because we wanted to reverse it back to make the oscillations. Yeah, basically there is here an illustration with a more detailed explanation, basically what we want. So we want that we arrive at the state in which protons arrive from the bulk wave faster than later urea. So what helps us is that the protons from all particles have the highest diffusion coefficient in water. They do not actually even diffuse. They are transmitted in a way that is a little bit similar to electric current. So it's a local concentration that is transmitted, but not every particular proton. And basically, after screening through many sorts of conditions, if you watch any of these 4 beads, I hope that the contrast on your screens is good enough to see that they actually oscillate. I will play the video once again. I was very excited when I have seen that because it was, as I believe, the first confirmation that this phenomenon actually happens in this exact system, more or less according to predictions. So I hope that everybody has seen oscillations. Then, when we tried to repeat the experiment under exactly the same conditions, which are presented here in the slide, is a certain concentration of urea and the buffer, certain pH. Wonderfully, at every repetition, we got completely different curves. So we are very sure that they oscillate. However, in many cases, the oscillations are irregular. In cases I will show later, they're just absent. Basically, the most wonderful to me is that it also reproduces when we resynthesize the particles. So in this sense, we can reproduce them, but we can never reproduce a trace. The trace, in fact, is different every time. So trying to understand what are the conditions in which the oscillations would be, yeah, I wouldn't mind, for example, if they all be like this green, nice curve, longer, more reproducible. We arrived at the situation when we found that it was actually a miracle that we had all found this region, particularly if we just change the concentration here by 5 millimolar. So it is, in this case, by 20%. We already arrived at the situation where it either does it or does not, and the oscillations become irregular. Then when we try to vary the other parameters, it appeared that, in fact, even changing the pH by 0.1 unit basically would, in one direction, completely kill them right away, and in another direction, it will shift them to this, what we call sort of bad oscillations, when it's a single wave and nothing happens further. However, at this standard pH, they reproduce very well. The system is less sensitive to only one parameter, which is the concentration of enzyme. It's also according to predictions in the theoretical papers. This is the only parameter that can vary in a quite broad region. And the most interesting thing that we have found is that as the oscillation depends on the local concentration of protons, it's not as unexpected, but basically the beads that are located nearby, such as these here, they exhibit collective oscillations. So in fact, because the local concentration of protons, let's say here in the middle of them, is exactly the same, they are absolutely synchronized at the same time, like these beads that are located in an isolated manner. They basically oscillate with a higher amplitude, but then they're not synchronized. And basically here, I think what lesson it gives to us is that we always need to take into account real systems and the problems that experimentalists face are in fact very different from the problems that theoreticians and computational scientists trying to solve. Basically, when I see a very nice model phase diagram, it suggests me a way on how to make my chemical system. However, the actual work to make it can just become a too difficult task and in fact unachievable, at least in a certain time frame. And it's remarkable that many of the discoveries in this field of the experimental oscillating systems were actually done by accident, so such as the BZ reaction. And I'm close to finishing. I just wanted to say that the autocatalysis, it can indeed be a curse for an engineer, but it can also play a good role. So in a recent publication, we used it basically to magnify differences. Absolutely obvious that autocatalysis magnifies difference. In chemistry, very often we want to tune the molecule in such way that the difference in inhibition of the enzyme will be as strong as possible, but then we need to vary molecular structure a lot. So in this exact case, when you try to do this on an autocatalytic target, you actually achieve it super easily because a slight difference will yield you very high kinetic differences. And why we need all of this? I believe in a future where we learn to make very smart biomolecular systems that will basically perform functions according to our design. So to achieve that, I believe a stronger coupling of theory and experiments is required. The biggest tragedy of chemistry is that we cannot set up rate constants as we want. And it is extremely difficult, as I've shown in this presentation, to find systems, to find the narrow regions on the phase diagrams of systems with interest behavior. So the possible way out of these problems that I see, and I'm potentially going to work in this direction, is to create systems that would sort of self-screen to the parameter space by using gradients or simply by let's say, automatized screening in which the system would by itself go through these many parameters, trying to find the place where I want it to be. So the real systems are always a problem, despite we imagine that in these particles, basically the stuff would be distributed homogeneously. If we dig really deep, if we have a good look, we always find homogeneities. And when in the case of linear systems, they don't do much to us, but it is exactly in the case of autocatalytic systems where they create very big problems. So I hope it was a little bit interesting and understandable, and I would like to finish with acknowledgements to my laboratory at the Radboud University, especially to my supervisor, Dr. Professor Huck, and the funders, the Dutch Science Organization and the Ministry of Education. And thank all of you for your attention.